So good morning guys, welcome to my channel, The Pharmacologist. So today we will be discussing about the topic clinical research and the most important part of clinical research, that is phase 0 or microdosing. So you might have heard about various phases of a clinical trial, that is means preclinical phase, then the clinical phase like phase 1, phase 2, phase 3 and phase 4. But there is a means new phase of clinical trial that is called as the phase 0 clinical trials. So why is it necessary, and why is it means important in the clinical trials? So let's discuss about the new phase, that is phase 0 clinical trial. So guys, if you see here, so the clinical trials has been divided into multiple phases. As you studied earlier, that is phase 1, then phase 2, phase 3 and phase 4. So there is a recent advancement in the scientific community. So the discussion is means very much deliberate about the new trial, that is phase 0 clinical trial. So remember guys, that I will be discussing those topics and those things that is highly essential for you to crack any type of clinical research interviews. So guys, this phase 0 means clinical trial is also called as microdosing. So this will be one question, that which phase of clinical trial is called as a microdosing. So you have to remember guys, the phase 0 of the clinical trial is called as microdosing. Why? Because here less than or equal to 1 by 100th of the pharmacological dose is given to the volunteers, and the dose should not exceed more than 100 microgram. So if you see here, the term microdosing can be well described by this thing, that is the dose will be given to the volunteers and that should be less than or equal to 1 by 100th of the pharmacological dose, and it should be not more than 100 microgram. So means earlier in the trials, in the conventional clinical trials, so the term microdosing or phase 0 was not there. In the recent advancement of research, so this phase 0 has been newly implemented. So this phase 0 is very crucial for the trials. Why? Because it reduces the time, it reduces the time, it saves a hell lot of money, and it also saves the participants from facing any type of side effects of the drug. So we can see here that it reduces the time. So if you see the clinical phases of a trial, so prior to the clinical phases, we have the preclinical testing or the preclinical trials. So in the preclinical trials, guys, generally the toxicological studies, the pharmacological aspect of the candidate molecule is studied on the animal models. So after the study is done, so IND is filed in the regulatory agency, that is investigational new drug. So the IND, it contains all the data like control, manufacture and chemistry. It is also called as CMC. So it is a part of regulatory approval process where the regulatory agency approves the IND data so that they can proceed further towards the clinical trial. So guys, As I said you, the preclinical study, the preclinical study, it happens on the animal models. So generally, rats, dogs, all these animals are used to study the pharmacologic and toxicological properties of the drug. And when we get a hint that the particular means chemical entity or the candidate molecule means can be tested on the human participants in the conventional clinical trials. It's after conduction of the preclinical studies, we were directly jumping into the phase 1. But what the problems we faced here is that the animal models are not same as that of the human participants or the human volunteers. A drug molecule or a candidate molecule may show that scientific and valid therapeutic effects or the PK parameters that is satisfactory in the animal models, but it may not work like in the human volunteers or the human participants. As we know, the chemistry and the physiology of the human body is far different than that of animal bodies. For that reason, when we directly proceeded from the preclinical studies to the phase 1 clinical trial, that is also called as the first in human use trials, so we faced a lot of consequences. That is, the study failed many times. Why? Because like I said, human bodies cannot be same as that of animal bodies. A drug molecule may be working good and may be showing satisfactory results in the human model, sorry, in the animal models, but it will not show satisfactory that what means outputs in the human beings. It may show severe toxicity or ADRs in the human populations. So these human the recruitment of human volunteers also, 15 to 20 healthy human participants will be studied here, and the PK parameters will be studied here, the pharmacodynamic parameters will be studied here. So all these studies required a lot of finance, and there was no guarantee that the study will pass. So to eradicate all these things like study failure, then toxicity and ADRs, then involving healthy participants without prior assessing the benefit risk and pharmacokinetic pharmacodynamic parameters and taking into consideration about the finance aspects. So it is actually not feasible to directly jump from the preclinical to phase 1. So the introduction of phase 0 clinical trials, the introduction of phase 0 clinical trials plays a vital role in creating a bridge between the animal studies and the phase 1 clinical trials. Got it, guys? So if you see, generally, as I said earlier, microdosing is a crucial part. For that reason, this study is also, the phase 0 study is also called as the exploratory investigational new drug study. So it's a very important, guys. It may be asked in the interview questions that which study is called as the exploratory investigational new drug study. So it is the phase 0 that is called as the exploratory IND study. It's very important. So this is a very that crucial question, that whether phase 0 studies involve any diagnostic or therapeutic intent. So the answer will be no. So there is no therapeutic or diagnostic intent. It is just used to study the molecule, that how the molecule will react in the human body in the initial stages. So here very limited human exposure is given, that is 10 to 15 healthy human participants are used for the study. But in rare cases, if it is an oncology trial, then they may include the cancer patients in the study. So generally healthy populations are used, but in the oncology trials, means patient populations can be used. So the duration of the study is very limited. That is, means for example, it may be 7 days, it may be also 1 day, and generally it is a single dose study. So this is the phase 0 study, and it is highly crucial so that the time, energy, and the side effects of the particular molecule means can be under control, and the finances will not be wasted if you are going for the phase 0 clinical trial. So that's all, guys. So this was a brief introduction to phase 0 clinical trial. Thank you so much. Have a nice day.